Thank you, Ajay, for the introduction. From my side, I welcome you all for today's session on complex portal. As Ajay pointed out earlier, so we'll be moving from proteins to complexes, and the resource for these macromolecule complex assemblies is our complex portal. So what are the complexes that we are talking about? Which are talking about the higher level assemblies which regulate protein function and stability. Okay, so let me walk you through this resource today. Okay, so Complex Portal is an open resource hosted at EMBL-EBI, and we stand for high quality manually curated macromolecule complexes, and we aim at providing unifying identifier to link knowledge on protein complexes across multiple resources. Okay, so let me define what is a macromolecule complex for a complex portal. So it could represent a stable set of two or more interacting molecules, typically containing at least one protein molecule, which have been demonstrated to form a biological function unit. So this clearly differentiates, like sometimes if you have an interaction data or a crystal structure having, two or three protein molecules, and unless it has a proven biological function as a whole unit together, we cannot consider them as complexes for complex portal entries. Okay, so mostly all the components of the complex portal entries are protein assemblies, but sometimes you can find small molecules and nucleic acids because they could have been, the reason that they are integral part of this complex. Okay, so let's try to address why do we need complex portal. So as I mentioned earlier, complex portal provides stable identifiers for complexes. So as a curator for a long time, and I've been working for molecular interactions database in Intact at EBI. So I curate a lot of molecular interactions. And in many cases, I would have got the reason to curate interaction wherein complex is one of the participant and where I have got evidence that it interacts with another protein B kind. So then I would need a complex portal identifier to say that these are the two entities that are interacting in this context. So likewise, the complex portal IDs are being used as a cross-reference in UniProt, Gene Ontology, Reactome, and Wiki Pathways. So Complex Portal also provides functional information on curated complexes, annotations from multiple resources. And also, most importantly, it can provide the training set for multiple machine learning algorithms for your prediction models. And also it can serve as gold standard for validation of your predicted complexes. Lastly, it could also help you to detect presence of macromolecule complexes in your set of protein-protein interaction network or list of proteins that you have. Okay, so how do we build macromolecule complexes? Basically, we follow the evidence and conclusion ontology, and there are specific terms that in that ontology helps us to define the level of proof that we have got for the particular complex. So at the higher level, so we would have got experimental evidence for all the components, means that some other biophysical or biochemical experimental data providing the presence of all the components of a complex in a single experimental setup, like column purification, crystal structure, electron microscopy, reconstitution, etc. So this is at the higher level. And there could be also collection of multiple weaker evidences, let's say more than a single co-immunoprecipitation, two hybrid interaction evidence, co-fractionation can also collectively add up as evidence level for complex portal entry. And also a complex can be inferred by orthology or parology by the curator. Okay, so now here I would like to define clearly what is set of interaction data and what is complex data. So interactions are basically you go by every individual case. So you look for individual evidences from and then you build data likewise. But complex portal entries, so you have to combine many pieces of evidences for a single complex, single entry of a complex. And for interactions, you can, each publication can be treated separately, like how much interaction data, how much evidences they have for multiple interaction detection methods can go separately. But for complexes, again, you have to bring multiple publications or multiple resources together. And interaction data can have evidence collected from scientific literature, or you can have the text mining or any other prediction models for interaction evidence. Complexes is mostly experimental evidence or inferred by the curator. Okay, so example of an interaction database is Intact, hosted at EMBL-EBI. So it deals mostly with the experimental data. And complex portal is a good example for complex portal, complexes database resource here. Okay, as I mentioned earlier, the complex portal entry can have many data resources. So it can fetch interaction evidence from Intact or IMAX Consortium or the collaborator partners, Campbell, Reactome, Wormbase, and PDB, Crystals, and so on. So, okay, so complex portal participants. So as I mentioned, like these assemblies can be entirely made up of proteins. So example here is this GARP complex. And it can have non-protein entities such as RNA molecules or DNA molecules. Like example here is a telosome. So here the telomeric DNA forms an integral part of the complex to deliver its function, right? So that's why. And you can have small molecules such as zinc as part of the complex, and sometimes subcomplexes are so it can be made up of subcomplexes and protein assemblies together. Okay, so coverage. So far, we have got more than 1,800 complexes curated for Homo sapiens. And we aim to cover at least, we want to cover 2,000 entries for human complexes, hoping to release them by first release of next year. But we have almost covered completely the complexome for yeast and E. coli. So we have published the first draft already. So we are keeping an eye for any new complexes in the space, and will be added eventually when we have data for them. And so there are many other species. We are also improving our Arabidopsis list. So this snake complex is entirely from my request. So basically, if you don't find organism or complex of your interest, you can mail us. I'll be providing the contact mail ID in the upcoming slides. You can write to us and we'll see to that it can be curated. Yeah. Okay, so now let us look at the data that will be captured for curated complex in complex portal. So this is a common example complex like hemoglobin A. Now let's see the details. Okay, so each complex, as I mentioned, I've been mentioning this. So it will have a unique identifier, so that provided by the curator, and there will be a recommended name again. So you have the species code and the level of experimental evidence available for each complexes. So in this case, it has got the highest level of evidence as a physical interaction evidence provided by Intact here. Okay, so then you will be able to see the details of the complex components in a complex viewer, so which has been developed in collaboration with Rapsler laboratory. So basically, it allows you to look at the details of all the components, and you can see the topology and stoichiometry and do much more. So then you have each node, the legend available for the representing the type of molecule that you are dealing with in the complexes. You can also have the description. You'll have the stoichiometric data wherever applicable, and also the biological role of all the components in the complex. Okay, so you'll also have details on the function of curated complex supported by Go ontology annotations at molecular function level and biological process level. And Wherever applicable, you'll have annotations fetched for like enzyme commission number, reaction, widget is also available. Recently, we have added that part, ligands, agonist and antagonist, and further details. Okay, so then you'll also be able to connect your complex with pathways, which is, and so mostly right now we have connected with reactome pathway, and you could see the widget and the link to the corresponding, pathway at reactome is also provided. Okay, so then further down, you'll be able to see properties, that assemblies, the physical properties, including the topology, weight, size, and cofactors, and cofactor data wherever applicable. And also you'll have the crystal cross-references from PDB. Okay, so then again, the most important part, which I would say is the expression in cell location. So the gene expression map is provided from GTEx project. It is fetched from GTEx project. And also the viewer that you can see is supported by Expression Atlas. So when we say about complexes, the components of the complexes are co-regulated. So the expression of all the components in a complex follow a pattern. So this needs to be taken care while you are doing some predictive models, predictive analysis to detect complexes in your network or your list of interactors that you have. Okay, so yeah, we also provide the details on cellular component location supported by Go. Further, we also bring the diseases and pathologies, and all these details will be supported by ontologies from EFO or Orphanet or HFO, wherever applicable. And if we have a drug target in Campbell, link is also provided with each complex. And you'll still have additional data from, here in this case, matrix DB, and you'll have the synonyms and, other details available. Okay, so now let us look on the complex viewer. So complex viewer is here to support us to view the details of, each complex components of the complexes. The details, which I mean is the binding domains, and between, and the link between which complex is bound to which participant is bound to which protein, so that interaction level, that linkage level. So such details are available, and you can be able to see them in the complex viewer. So basically, you can expand all the components of the complexes. You have an option called expand all. So here is another example for complex wherein you have the binding domains. So that has been linked with each of the participant of the complex, and you have the mapping in which range is binding to which protein and etc. So, and you can view further to see that, zoomed in to see that the linking or the binding domains are being mapped to the amino acid sequence level at the proteins. So basically, we run the protein updates every time whenever UniProt does release, whenever it has a new release, and so that all the sequence data and your binding domains data are always in sync or up to date with the latest release of UniProt. Okay, yeah. And then, so our complexes, as I mentioned earlier, the complex can be considered as a training set for machine learning models. So recently, we have been successful in collaborating with, in generating a project in collaboration with, Kevin Drew Lab, which will be at the University of Illinois, which will be extending the HUMAP, which is a global compendium of human multiprotein assemblies. And we have a collaborator from University of Edinburgh, Georg, and he'll be working towards identifying high-confidence protein complexes by validating and refining using protein co-expression analysis. Okay, so then at the last stage, EMBL-EBI will be enriching the inferred high-confidence complexes with structural and functional data and creating complex portal entries. For the stage one, we have already provided our complexes for Kevin Drew Lab to do their machine learning model. And so they are ready with their first set of complexes. I think they have generated more than 10,000 complexes, but they want to do some further refinement. And hopefully in next year, like we'll have some new entries in complex portal based on this machine learning based prediction models. Yeah. And so what can we do with these complex portal entries, basically? So there is a nice publication by Ludo et al., and I have shared the PubMed ID here. So basically, it gives a nice framework for predicting complexes. So starting from using a clustering algorithm to identify highly connected nodes, because the proteins that are, that forms a macromolecule complex share a common function, and they tend to be associated together, and also they are interconnected among themselves than compared to the rest of the nodes in a network. So that could be a good starting point for detecting like highly interconnected modules in a network to detect for presence of complexes. And yeah, so then you have to compare the expression levels or the stability of the proteins, because all the components of a single complex, they are co-regulated. So means that you need to have a look at their status of expression data. And then most, I think, complexes have a common, I mean, they have common evolution. I mean, so they have the common function. So then you have to look at the protein-protein complex evolution, conservation versus extension of protein function. Okay. So if you are doing your network analysis in Cytoscape, and our complex portal entries are available in Klugo for you to check, I mean, detect presence of any complexes in your list of participants. So basically, it is specifically available for, Klugo version 2.5.8. Other versions do not support complex portal ontology. I mean, sorry, as a resource. So so in Klugo, you have to give your list of input as a uniprot accession numbers, or select a pre-existing network module for analysis. Then you have to select our complex portal for ontologies, and you can do some advanced, selections in, pathway option. I mean, I can explain you in the slide. Okay, yeah, here is the details further. Yeah, so in Klugo, the version 2.5.8, and I have given my list of uniprot accession numbers in the search box. Actually, you can you can import using a file, or you can just directly give the inputs in the search box, or you can, if you have a network, you can select a module from your network and ask Klugo to fetch it from the network using this option. Okay, so then you have the visual style option. Basically, by default, it go for groups. You can change it to significance. That doesn't matter. Like, it's still, you'll be able to get complexes. But the crucial stage is the ontologies and pathways. As I mentioned, like you have to select complexes, but the most critical part is like you have to get the latest ontology, I mean, latest files from complex portal. So basically from Klugo, you can do enrichment for multiple resources like Go, Reactome, KEGG, or many other resources. But then you have to ensure that you have updated all the ontologies. So whenever you select an ontology, make sure that you have the latest version. So you have to click on this update ontology option to get the latest version for your analysis, up-to-date analysis. So then the downstream, you'll have advanced term and pathway selection options, wherein you can select the Go tree level, which is like minimum of 3 can give you the prediction, I mean, good standard of predicted complexes. Likewise, for the Go term pathway selection, again, the number of genes that you want to be considered for a complex to be predicted. So you can give minimum of 3, and higher than that will have a good relation, I mean, good scope of finding complex. Okay, so for my analysis, like I gave my list here, and this is the outcome of complex and from complex portal entries that I've got. And in Klugo, you'll be able to save the result, and you can save the result in Excel, and you'll be export the image file as PNG file. And yeah, here you can see the complex portal complexes, like, the corresponding complexes being listed here. And when you slide through this, you'll be able to see the list of all the components that are being, found for each complex and the relative number of scores given for each number. Okay, so why from networks it is like, from modules, it is easy to predict complexes. Let me give you an example. So there is this property of, clustering. So the network shows clustering if a probability of pair of nodes being adjacent is higher when the two nodes have a common neighbor. So basically, complex components, if basically they share a common function, so they have to bind together. So they do a lot of interconnectivity and they appear as a cluster or module in a network. So to your right side, you see the ubiquitin proteasome system network. And so this is basically a set of, so my previous manager generated this network. So when he was doing his postdoc, I bet, so he generated this network from set of neurodegenerative proteins. And I think the color code is based on the cellular component, their location on the cellular component. So as you can see, there are like lot of nodes which lot of proteins which do not have higher connectivity on the external part of your hairball, but there is like a lot of interconnections happening here in the center of the network, right? So there is this one interesting part. So this cluster, which has the common color code, and it has a lot of connections between here. So when we actually looked into the details of these components, they are part of the proteasome complex cluster. So this is the relation between protein clustering and those functional modules being could be part of like a macromolecule assembly. Okay, so if you are in exploratory mode and if you want to look for all the complexes in complex portal or search for a particular complex in complex portal, so that is possible. And this is the web page, and you have the search box wherein you have the search examples. Basically, you can try to search via gene names, or if you have if you know the uniprot accession number of a particular protein, you can search by it. If you know the complex portal ID, complex ID for a complex, yeah, that also helps. Complex names, and you can search for Go ontologies as well, or you can provide list of accession numbers together to look for a complex. Okay, so then you can do a wildcard search to fetch all the complexes from complex portal. So then you'll get all the complexes, and you'll find filters in the left appearing at the species level, appearing at the biological role type or molecular type. So you can filter accordingly and look for the complexes. Or you can access programmatically our complex portal data, or you can go for species specific complexes by using this organisms tab. That's also available. And you can download the details of the complex portal entries in multiple formats. Basically, we support, XML format. There's a complex tab available, and also in MIJSON format. Okay, so with this, I thank all our contributors, the intact and complex portal team, past and present members, our collaborators, SGD, Flybase, and other contributors, the UniPro team, and all our funders. And this is our complex portal mail ID. If you have any questions, you can mail to us. So with this, I complete my presentation. I'll be happy to take any questions from you.
Thank you very much, Kalpana, for the webinar today. And thanks everyone for joining in. So we are now in the Q&A session, so feel free to type in your questions now. So there's one question coming in. From such a huge network as you have shown in ubiquitin proteasome system network, how do we choose any one cluster? What factors we usually take into account while choosing any one cluster from such a huge network?
Okay, so I'm sorry. So basically, so if you have generated a network, so then within within Go, okay, okay. So in Klugo, while you are using the Klugo app, so first, if you had generated network using in Cytoscape, so basically within the network, you can actually select like you can select a network within the, in the cluster, within the cluster, and ask Klugo to, using this option from network. If you click on this icon, so it will allow you to choose the cluster within the network and use this folder like option to fetch the interactors, I mean IDs of your, the proteins that is found in your cluster, so that it'll consider for analysis. So basically you have to define the species and the type of ID before you actually select the cluster, so that it doesn't confuse. But there will be a default option. But if you have to be really specific, like if you are giving the uniprot accession numbers as, as the identifiers, then you have to select it earlier. So right now I do not have I do not have an open session, but we will be planning a lot of training modules in the next year, and I can really show you how to select a module from the cluster. So basically when we say a cluster, because they are tightly interconnected modules, so they have more interaction. And so when you calculate, when the algorithm calculates, it will start from it will start from the highest interacting cluster and see if the components are basically matching to any of these complex portal entries. So that is the idea. But the predicted models of, if you want to predict any functional module within a network, so there are several applications like M-code. So this application will allow you to find the interconnected module in your network. And so basically it starts with the highest node. I mean, so having highest degree of connections within this clusters, and then it will start building from that node to the extend the cluster. So then you can use that as a starting point. So I have not predefined a network right now to show you, but I'm sorry, but we can do it if you join us for the training modules later. Yeah.
Okay. Thanks, Kalpana. So next question, this was a wonderful webinar. If a protein is existing into oligomeric assemblies, how can we predict the specific functions of different oligomers at different times in cell? Any softwares available to predict the same?
So I presently do not have a direct answer to this question, because this oligomerization, it depends on the context, and I'm sorry that I do not have a direct answer. Yeah, sorry.
Yeah. So I think the next question is continuation of that one. So different oligomeric transitions can perform different function. Can we track those functions by some softwares?
So at least like I don't have any software, but at least in complex portal we provide the stoichiometry, and in intact data whenever we have the oligomers, we have these oligomerization data or self interactions, we do capture them, but I do not remember any softwares that, well, I have not come across any of that sort. Yeah.
Okay, thank you. So in the next question, so algorithms that create clusters, are they based on physical interactions?
Okay, so then the network will be generated. It depends on the network actually you selected. So you can generate your network based on physical evidence, or you can do that by predictive models. But the strength of the interaction will be higher when it is coming from an experimental data, right? So we have we have generated a Cytoscape application, so wherein we can intact Cytoscape app, wherein we can fetch interactors from intact database, and we have something called the molecular interaction score filter. So that will filter for the high, so this filter allows us to restrict the interactions which can pass about a certain threshold of interaction strength. Let us say, so there's this strength of the interaction is calculated by multiple pieces of evidences. The more stronger the evidence, the more is the strength of an existing interaction, and you can actually set a threshold so that you get only the high confidence interactors, and then when you, if those interactors are found to be expressed in a common tissue, say a neuron, and from that, so you generate a new set of network with those expressed in a particular tissue, because all the complex components are co-regulated. So either they are expressed in same tissue or regulated in the same tissue. So their expression is it's highly regulated. So you need to consider that data. And then from that, you can pass this module for presence of, for EMCode algorithm or cluster maker algorithm in Cytoscape, or you can try to bring that module for enrichment in complex portal in Glue as we did. So that's a possibility. Yeah.
Okay, thank you. There's another question. I think Sucharita is answering that one. So the question was about how complex portal is different from UniProt and SwissProt.
Yes. So basically, it is the difference of proteins and assemblies of proteins. So it is not just assemblies of proteins. The proteins assembly should have a common. Should be a functional unit and should have like experimental evidence on the functionality of that as a common unit, the entire unit. So you can consider complex portal at the higher level resource of proteins like like UniProt, but it is at the higher level. It talks about higher level assemblies. Yeah.
Okay. So the next question is: does the complex portal give all the data available in Reactome? Is there a link between complex portal and Reactome?
Of course, there is always a transition. So we share our data with Reactome, and so every complex entry will have its corresponding Reactome pathway widget integrated with complex portal in the data you see. So yeah.
Yeah. Thank you. There are no further questions yet. We still have time. You can send more questions in here in the Q&A box. And while we are waiting for questions, I would like to tell you that the molecular building blocks of series is still running, and we have 2 more webinars coming up later in the following weeks. So one is about human cell atlas, and the other one is about metabolites. So this is happening on 22nd November and 29th November. Okay, so we have another question. Could you kindly let us know the softwares which can predict oligomeric nature? But I think this you are already answered, Kalpana. I think there is.
So I'm not the right person to answer. I don't have the right answer.
Yeah, you can write to EMBL-EBI help desk. I can send that help desk email in my email that I'll send it to you by tomorrow, so you can contact them. Okay, next ones. Could you give us some example of how it can be used if we are studying on Fragile X syndrome?
So you, I'm assuming that you're having, so Fragile X syndrome. So you are picking up a set of proteins that are known to be associated with Fragile X syndrome. So if you want to predict presence of complexes in it, so then the first stage, I would request you to find if any of your proteins have interconnectivity. So basically you fetch your give your input data in Cytoscape and look at the network and see and check the highly interconnected network modules in M-code application or cluster maker. And then you have to import or bring in a particular expression data for a particular tissue. I don't know, the Fragile X syndrome. I mean, so which disease, I mean, which organ, which tissue it affects mostly. So you check the expression level of your module in that, particular tissue. And then from that, you generate a new network and then check for, complexes. So that is one way of looking for complexes in your list of proteins. Yeah.
Okay, thank you. In protein-protein interaction network, is there a set confidence score that helps you in selecting the network?
Yes, so we have in Intact Cytoscape app. So Intact Intact is a molecular interactions database, and from our resource we have generated a Cytoscape specific app. And so when you use that app to fetch interactions from Intact data, so the app will allow you, give you an option to filter out or bring only the high confidence interactions data. So there's an filter level at the edge tab, so that you can do it. So the minimum threshold, I would say, is like 0.5. So the entire range runs from 0 to 1, and 0.5 would be the normal threshold for bringing in high confidence interaction. So you can go much higher than this as well. Yeah.
Okay, thank you. Thank you for this webinar. Can this portal be used to check the interactions of proteins with peptides?
No, I mean, you are looking for complexes from complex portal, but if you're looking for predicting interactions with peptides, and maybe AlphaFold could be the option, because there you find there you have the option of finding the pocket or the interfaces, binding interfaces. So then that should be, I think that would be the relevant resource. Yeah.
Okay, thank you. The next question is, I think, again about the p-values. I think the question aims to ask that, is there a way to sort the network that you predict, like top 20 networks for a complex?
Okay, so if you are starting from a network, so if you try to filter, if you try to find modules via M-code application, so it gives you in the hierarchy, like the top scored network in the top, and based on in that order, it actually generates modules like that. So that's an option. Yeah.
Does the portal provide details for microbial proteins, for example, for drug target identification for antibiotic-resistant bacteria?
So that could be, so we have, we have the call a complexome as a model complexome for this microorganisms. And wherever we have the targets from Campbell, so we have integrated with the complexes there. So you can fetch them programmatically or by, XML files. Yeah.
Okay, thank you. Are there any more questions? In the chat, I have typed in the link to Intact, which Kalpana mentioned a few seconds back. You can explore that to find a resource for molecular interaction database.
Yes, so I have given the link is also available here. Yeah. Once this slides in papers.
Yeah, somebody is asking, while using the portal, if we have any query, where we can send our query.
Okay, so you can you can use the common feedback mail that EMBL provides you, common feedback link that EBI provides, or also you can mail us to complexportal@ebi.ac.uk. Yeah, you can request for new complexes or any suggestions, feedback that you want to see us. Yeah, you can use this mail ID to reach us.